welcome to this explainer. Today, we're diving deep into the neural language of the retina. We're going to explore how this incredible piece of tissue operates, not like some simple biological window, but much, much more like a highly sophisticated mini-brain. If you've ever found yourself wondering how tiny photons of light are translated into the rich, complex visual world you experience every single second of your waking life, well, you are definitely in the right place. Let's get into it. So what actually happens when light hits your eye? A lot of people just assume the eye is basically a camera, right? Passively capturing light, using a lens to focus an image onto a blank biological canvas. But today, we really need to question that simple assumption, because as you'll see, the reality is far more dynamic. Actually, scratch that whole camera idea. The retina is absolutely not just a piece of camera film. It is a highly sophisticated neural processor. Before an image ever even reaches your optic nerve, your retina has already performed a staggering amount of computation. It compares, it tweaks, it modifies visual data. The retina is literally a part of your central nervous system, performing the initial, heavy-lifting integration of visual information before that data ever even leaves the eyeball. To make sense of this biological circuit board, we've got 5 key items on our agenda today. 1. The retina as a brain. 2. Translating light to electricity. 3. Modulating the visual signal. 4. Synapses and clinical maps. And 5. The final neural output. All right, starting with Section 1, the retina as a brain. Let's meet our cast of characters. Rather than just a flat, simple sheet of light-sensitive detectors, the retina is packed with an ecosystem of interconnected neurons and glial cells, all working in perfect unison. You've got your principal neural elements, the photoreceptors, which are your rods and cones, but then you've also got bipolar cells, horizontal cells, amacrine cells, and ganglion cells. Oh, and we absolutely can't forget the Mueller glia. They provide the vital structural and metabolic support across the entire retinal thickness to keep this whole complex operation running smoothly. Moving right along to Section 2, translating light to electricity, or what we can call the photoreceptor paradox. This is where the timeline really kicks off. Okay, here is a genuinely bizarre biological plot twist. In most normal sensory neurons across your entire body, stimulation causes the cell to depolarize. It essentially turns the cell on. But retinal photoreceptors, they do the exact opposite. In total darkness, they kind of just sit there at roughly negative 40 millivolts. But when a photon of light strikes them, they actually hyperpolarize, dropping all the way down to about negative 70 millivolts. So weirdly enough, they essentially turn off in the presence of light. Now, this sudden drop in voltage doesn't just happen by magic. It's the result of a highly precise biochemical domino effect that we call the phototransduction cascade. It kicks off the exact millisecond a photon hits the visual pigment, rhodopsin. That tips the first domino, a G protein called transducin. Transducin then knocks into phosphodiesterase, which lowers the concentration of a molecule called cGMP. And as those cGMP levels drop, the sodium channels in the cell just snap shut, causing that hyperpolarization we just talked about. Just to put that into perspective, think about the sheer efficiency of this cascade. A single activated rhodopsin molecule can activate up to 100 transducin molecules. This cascading process creates a massive, built-in biological amplification factor. It allows your retina to take an infinitesimally tiny amount of light energy, literally a single photon, and ramp it up into a robust, undeniable cellular response. But of course, that electrical shift inside the cell has to be communicated to the rest of the retinal network, right? Enter glutamate, an excitatory neurotransmitter. Because light changes the electrical state of the photoreceptor, it fundamentally alters how much glutamate is released to the synapse. So the bipolar and horizontal cell networks are quite literally reading these shifting chemical inputs. At this point, the visual information has been completely chemically encoded. Which brings us to Section 3, modulating the visual signal, focusing on the lateral processors. Light is now an electrical and chemical signal, and the retina really starts acting like a circuit board. At first glance, we have this straightforward vertical pathway, right? Photoreceptors to bipolar cells and then straight down to the ganglion cells. But listen, if the retina only relied on this vertical relay, our vision would be incredibly basic. It would pretty much only transmit simple brightness. But human vision is dynamic. It operates across huge ranges of lighting and requires ultra-precise contrast and timing. The reality of our vision demands a much, much more complex biological circuit. So, superimposed right on top of that vertical pathway are these amazing lateral processing chips,
are horizontal and amacrine cells. Horizontal cells hang out in the outer plexiform layer, constantly comparing inputs from neighboring photoreceptors. Why? Because your brain cares way more about edges, borders, and contrast than it does about just absolute brightness. Meanwhile, amacrine cells, which are located closer to the inner retinal surface, are busy tweaking and modifying these signals based on timing and spatial relationships before the ganglion cells can send anything off to the brain.
And get this, because this neural network is firing on all cylinders, it demands a massive amount of energy. That's where neurovascular coupling comes into play. As these retinal neurons work harder and harder to process a visual scene, they actually release substances like nitric oxide, which acts as a vasodilator. That means the act of visual processing physically alters local blood flow. It ensures that the retina's metabolic supply dynamically matches its functional demand in real time. How cool is that? All right, Section 4, Synapses and Clinical Maps. How does doctors actually map these microscopic synaptic pathways in a living, breathing patient? Well, when clinicians want to objectively map these electrical responses, they use a full-field electroretinogram, or ERG, and they look for very distinct waves. The initial A wave reflects those photoreceptors hyperpolarizing. The subsequent B wave comes primarily from the depolarizing bipolar cells, and those subtle little oscillatory potentials you see, those are the amacrine cells hard at work. So an ERG is literally a real-time electrical map of the different processing stages we've been talking about. On top of that, we can use what's called a pattern ERG to isolate even more specific stages. For example, the P50 component tells us all about cone-driven responses, while the N95 component originates predominantly from the macular ganglion cells. Clinically, this is a total game changer. If a patient is having vision issues, doctors can pinpoint exactly where this biological circuit is glitching without ever having to touch the eye. They can literally see if the ganglion cells are struggling while the photoreceptors are totally fine. Zooming out just a bit, we also see this profound anatomical specialization depending on exactly where you look in the eye. Just compare the fovea to the peripheral retinal. The fovea is your ultra-high-resolution center. It's densely packed with cones and totally optimized for daylight acuity. The inner retinal layers are actually physically pushed aside there, just to let light hit those cones directly. The peripheral retina, on the other hand, is a stark contrast. It's dominated by rods, perfectly optimized for high sensitivity and dim light vision. Different functions demand entirely different physical circuitry. And finally, we arrive at Section 5, the final neural output. Let's trace this signal from the retina straight to the brain. Leaving the inner plexiform layer, all of this beautifully refined, heavily edited data is handed off to the ganglion cells. These are the cells that encode the absolute final neural output. Their incredibly long axons travel through the nerve fiber layer all across the retina, eventually converging, literally gathering together, at a single exit point, the optic nerve head. From there, they bundle up to form the optic nerve, transmitting all that computed data out of the eye and over much greater distances to the central visual pathway. Which brings us to, really, the absolute core thesis of this whole explainer. The optic nerve does not transmit a photographic copy of the retinal image. It transmits the result of retinal computation. Think about it. The light entered the eye as raw physical photons. But what actually leaves the eye? It's been entirely transformed by a biological computer. It's been amplified, compared, inhibited, and intricately encoded. And honestly, that is the staggering achievement of retinal processing. It's a massive integration of complex data happening in just a fraction of a millimeter of neural tissue. But it leaves us with a pretty provocative thought. If this much editing, tweaking, and processing happens before the image even leaves the eyeball, what other processing secrets does the rest of the visual pathway hold once that signal actually hits the brain? Just how deep does the rabbit hole of human vision truly go? Thank you so much for exploring the amazing neural circuitry of the retina with us today. Keep wondering and keep learning.